Thank you, Meredith, for kicking us off and for and as always, your outstanding job of organizing and putting things together. It's my pleasure to introduce today's Club EvMed speaker, Dr. Aida Andres. Dr. Andres is a quintessential evolutionary medicine scientist whose work focuses on how organisms adapt to their environment. She analyzes genomes, both modern and ancient, to infer how natural selection mediates genetic adaptations. She's a pedigreed cross-disciplinary scientist, getting her PhD working on primate comparative genomics at the Universitat Pompeu Fabra in Barcelona, Spain. This was followed by 2 postdoctoral positions in human population genomics, first at Cornell University and then at the National Human Genome Research Institute at the NIH in the United States. She launched her faculty career by founding a new group on population genetics at the Max Planck Institute for Evolutionary Anthropology before accepting her current position at UCL Genetics Institute in 2017. Her work has focused on humans and endangered primates, studying their ability to adapt to diverse and rapidly changing environments. Her group tackles these questions through the application of genomics, population genetic techniques to make inferences on the history of selected alleles, and functional annotation to infer the consequences in present-day phenotypes of previously adaptive alleles. The title of Dr. Andres' talk today is Genetic Adaptation to Zoonotic Viruses. How did chimpanzees adapt to SIV, HIV infections? Aida, the floor is yours.
Thank you very much. Thank you everybody for coming to my talk. I can see some colleagues and friends, and some of which I haven't seen in a long time, so it's really nice to see you here. But thank you for coming, also to those that I don't know personally. Yeah, so I've prepared a not very long talk with the idea that there will be some discussion. I was telling Meredith I'm coming down with something, so if I'm a bit slow, you have to, it's not COVID, but you have to excuse me if I'm a bit slow, hopefully not too much. I also wanted to say there are 2 PhD students who have worked with me on this project in the audience, so hopefully I can answer your questions, but I'm sure they will be better than me at answering some of them. So I'll introduce them later on, but they're here. Let me share my screen. And I hope this works well. Yes, perfect.
Looks great.
Perfect, thank you. Yeah, so I'm gonna tell you a little bit about, so first I wanted to thank all the organizers for inviting me to present here. It's really nice. I'm really an evolutionary biologist, population geneticist, but I'm very interested in the effect that evolution has on important phenotypes and on health phenotypes and on disease. So it's really very nice for me to be able to talk to you. And I'm going to talk about genetic adaptations to zoonotic viruses, focusing on adaptations to SIV in chimpanzee. This is a subject that we come to from the very evolutionary side of it. And it has made me think more about how we can use evolutionary biology in order to better understand adaptation to viruses and zoonotic viruses in particular, which are obviously a huge issue. They have been a very big issue for a long time, but now they are just on the news every day. Yeah, so let me go right into it. So genetic adaptation, I would say, it's a really very important process. It just drives all differentiation that we see in nature, or at least the one that that allows you to survive where it is that you're living. I would argue, it's responsible for the huge variety of diversity in life that we have, but it's not only responsible for differences between species and at the very big level, but also within species, cross populations, they adapt to their very particular environment And one of the important things about genetic adaptation is that it allows populations to adapt to changing environment. So when their environment changes, they are able to adapt and continue living, continue thriving in that environment that has changed. Now, there are many environmental factors that are very important that we know both in humans and in other species that have driven genetic changes. And those include, for example, climate, and those include, for example, diet. But a very important one is obviously pathogens, right? And viruses are one type of pathogen. And pathogens are interesting for a number of reasons. One of them is they are clearly a very strong selective force because they can kill you. But also, they sometimes are completely new, right? And this is the case of zoonotic pathogens. So a zoonotic virus is a virus that a particular species or population has never seen before, cannot respond to effectively. It's transmitted from another species. And then all of a sudden, you have a new pathogen that is often very, very highly pathogenic because your immunity doesn't know how to deal with it, that you have, as a population or as a species, you have to adapt to in order to survive, to reduce its pathogenicity. And this is something that one very, very commonly sees, and is that zoonotic pathogens are very, very pathogenic as a start in a naive population. And then with time, populations and species adapt to them. And I would like to use evolutionary genetics in order to understand how these processes happen, because that not only help us understand evolutionary processes and adaptation, but also allow us to identify which are the host genes that are very important in order to reduce the pathogenicity of a virus that maybe kills another species that is naive to the virus and it doesn't kill a species that has adapted to it. Which are those genes that are so important that they can kind of keep a virus under control? So in my group, we studied the genetic basis of genetic adaptation. I would have to say most of our work is actually on humans. So we study how humans have adapted to the different environments that we live in, and what are the consequences in modern populations of those past adaptations. We use ancient genomes, we use modern genomes. And I was very tempted to tell you about this today, but in the end, and after conversations, I've decided to tell you about SIV, which has to do with our work on non-human primates. So we also work on endangered primates to try to understand how they adapt to the environment. And I'm gonna talk specifically about our work on chimpanzees. And this is, as I said before, the work on, you will see the story, the reason why we're interested in SIV comes from the very evolutionary side of it. So these are the three people that have been very, very strongly involved in the work that I'm going to present. Josh Smith, who was a postdoc in my group for several years, and he's now back in in Australia, where he's from. Harvinder Pawar, who was a master's student in my group and who's now a PhD student in Barcelona and who's here in the call, I believe. And Harrison Ostrich, who's a PhD student in my group, who I also believe is in the call, who has been working on this also in the most recent years. So I'm not an SIV expert nor an HIV expert, but we've been learning a lot about these viruses through this work. So a lot of the idea of working with chimpanzees is the fact that, well, we're interested in human evolution, and humans, of course, if you put them in their evolutionary context, the closest living relatives, our closest living relatives are the chimpanzees or the bonobos. We are extremely similar in most ways. So we're only 1% divergent at the genome level, which is really very, very low. In many other species, that's the level of divergence that you see having between subspecies, for example. We have very similar physiology, we have very similar behavior, and we have very similar pathogens. And actually, most of the pathogens can go back and forth between the two species without many problems. And actually, HIV is a case of SIV, the Simian Immunodeficiency Viruses, that jumped from chimpanzees and other species, but I'm going to focus on the jump from chimpanzees into modern humans, and has created a pandemic that is one of the worst in human history. And that is still a huge problem in many places in the world, including in Africa. And that it seems some theories suggest that might be responsible for some of the or the fact that humans cannot control the virus might be responsible maybe for new strains of even SARS-CoV-2. What is clear is that it's still an ongoing problem. It's a really complicated virus. We don't understand it very well. But it's also clear that even with all the complications, it can jump from chimpanzees to humans, and certainly can jump from humans to chimpanzees, right? So chimpanzees have SIV, and in humans we call it HIV, but it's a very, very similar virus. So chimpanzees can help us understand humans, not only human evolution, but also human physiology from the evolutionary point of view. Now we're starting to have population genetic data for many of the species, which allow us to do really very, very high quality evolutionary work in these species and compare that to humans. In humans, we have thousands and thousands of genomes now, but for many of these species, we're starting to have numbers that allow us to do proper evolutionary population genetics. So chimpanzees, there's not only one subspecies, there are 4 recognized subspecies of chimpanzees. The Western, Nigeria, Cameroon, Central, and Eastern, and this is just the names come from where they are in the whole range. They live in this very restricted area of sub-Saharan Africa. Compared to humans, it's really, really restricted. Now, if you look at the relationship between these subspecies, we have the Central and the Eastern subspecies, which are these 2 that live here. They have a recent common ancestor around 100,000 years ago, maybe. They have relatively large effective population sizes. Then there's the divergence with the second branch, which is the Western and the Nigeria Cameroon. So they are on the Western side of the range. They have very, very small effective population sizes. And they've diverged from Eastern and Central around 400,000 years ago or something, we believe. So I tend to say often that Eastern and Central are differentiated as human populations, and Western Nigeria, Cameroon from Eastern and Central are comparable to human to Neanderthals. This is work that has been done with 65 genomes from individuals from African sanctuaries, and everything that I'm going to tell you about today is based on this data, even though we have larger numbers of genomes now, but we're still analyzing them. So in terms of differentiation between the subspecies, as I was saying, Central and Eastern are as differentiated genetically as 2 human populations. Nigeria, Cameroon, and Western are more highly differentiated. And Westerns have, because they have this very, very small effective population size, they have very large effects of drift, and that's why they are very differentiated with everyone else. The important thing for me that you remember is that there are these 2 differentiated clades that have been apart for some hundreds of thousands of years. So we came into this trying to ask whether, do we find any evidence of differential local adaptation between these communities? So, sorry, not between these communities, between these subspecies. They live in this relatively restricted part of the world, but they have been differentiated for some time. We know they're genetically differentiated. Have they adapted differentially? And that's important for many reasons, and one of them is for conservation purposes, obviously. And then the next question that we have that I actually cannot see because, okay, I can see now, is whether this differentiation is due to different selective pressures in the different subspecies, or there are shared selective pressures that they are adapting to in different ways. So Josh was the person who was leading this work, and I won't go into much detail because this is published work, but I will tell you what I think that it's important to follow on the SIV response. So in order to study differential adaptation between the 4 subspecies, we took a relatively simple approach of studying allele frequency differentiation, and we use PBS, the population branch statistic. This is a relatively simple statistic that just compares allele frequencies between 3 populations and asks how long the branch of differentiation is in any of them. The PBS was initially designed for 3 species, and what Josh did is to extend it to 4 species because, 4 populations, because we have 4 subspecies, right? And the idea here is, as I said, is how long is this branch in terms of allele frequency change? So this is a statistic that is done per SNP. So what you would have in a normal SNP that behaves as you would expect under neutrality is that first, it follows the known phylogeny in terms of allele frequency differentiation. So Central and Eastern will be more close in allele frequency than they are to Western and chimpanzees. And then you have some length of this branch that depends on the amount of drift, so differentiation of this population. Now, if you have a very, very short branch, that means that particular SNP has changed very little in allele frequency, for example, because it's under purifying selection. So it's under constraint and it cannot change. If you have a SNP that, or a variant that looks like this, what you have is it has a very large allele frequency differentiation, specifically in the easterns. And this is exactly what you would expect if this site has been under positive selection in the eastern and has mediated positive selection, so differentiation in the easterns. So we use this method that has been used really, it's very used very commonly. We used it in order to study population-specific or subspecies-specific adaptations in each of the 4 subspecies of chimpanzees. And just very, very quickly, what I can tell you, do we find evidence of local adaptation? Yes, we do. But we have very strong evidence of local adaptation in the eastern chimpanzees only. So we know that there is positive selection in the other subspecies, but we know that we have much stronger evidence in the eastern chimpanzees. This was really a big surprise to us. And we spent a lot of time as population geneticists that we spent a lot of time trying to show that this is really local adaptation. This is not because of drift. This is not background selection. We really cannot explain the signatures in the absence of local adaptation. What was really surprising, as I said, is that it was specifically the easterns that showed this stronger evidence of selection. And it was very difficult for us to understand why that might be. So then, in order to try to understand why the Westerns would be so different, we asked, what are the selective pressures? Can we, based on the genomic data, identify the selective pressures that might be responsible for these adaptations? So we took the approach of studying whether the SNPs or the genes that show signatures of positive selection are enriched in any particular category that would help us understand these signatures. So putatively selected sites in easterns are enriched in sites with predicted regulatory function. So what this is telling us is that there was some change in regulation, but not in strongly conserved sites. So what this suggests is that whatever functions are changing are not highly constrained. These must be functions that change across species, but we believe that they are enriched in regulatory function. And there were several immune-related categories, and I'm just giving you the ones that had the strongest enrichment. And you can see that they are all immune-related. And this made us think that maybe there was some pathogen driving this very strong adaptation in the easterns. Now, Josh started thinking about this, and the first thing that the very first thing that he wanted to check was SIV. And SIV, why? Well, it's a very, very important pathogen in chimpanzees. So the Simian Immunodeficiency Virus is a lentivirus that infects over 40 species of African primates, and it's a precursor of HIV, which results in AIDS in humans, as I said before. It is really highly recombinogenic and highly zoonotic, and it has been transmitted across species in Africa dozens of times. So it really jumps species relatively easily among those species that are infected. Now, non-African primates are, and including humans, non-African primates and humans, because humans are an African primate in a way, are naive to the virus. So they have never seen it before. They are not natural hosts, and then they will go and develop immunodeficiency. In humans, we have several groups of HIV-1 and HIV-2, and they have different origins, right? So the one thing important here is the HIV-1, the main HIV-1 groups were transmitted from a central chimpanzees into humans, but there are additional groups that haven't become pandemic, but that have also jumped from other species into humans. They just haven't become so common. And chimpanzees, as I said, centrals are the origin of the human HIV-1 pandemic. So the chimpanzees are interesting because when you can infect any of the subspecies of chimpanzees with SIV and with HIV, I believe. And they are what we in the lab we call semi-natural hosts. They have been in contact with the virus in nature for a long time. They don't typically have AIDS in the same way as humans do, but they are not perfectly natural hosts like this species of African primates that have been in contact with the virus for a really, really long time. So I don't know, vervet monkeys, for example, they have their own strains of SIV and they really are not bothered by it. It doesn't seem to reduce the fitness. Chimpanzees are not there. Chimpanzees have some physical effects. Some people believe that they have some effects on fitness, but it's true that they are not as affected by it as humans. They don't develop AIDS in the way that humans do. Though I have to say this is a subject that has changed a little bit. So I would say that for a long time we thought that the chimpanzees didn't really have any health consequences, and now I think that the view is shifting toward considering them semi-natural hosts, maybe. Now, what is interesting about SIV is that it does not infect in nature all of the subspecies of chimpanzees. It is seen in nature in certain and eastern chimpanzees. It has never been observed in Western Nigeria, Cameroon, and I know that people have tried very hard to find it, and they haven't. So we don't believe that Western Nigeria, Cameroon are infected in nature. You can infect them in the lab, but we don't think that they are infected in nature. So this is interesting because because I told you that Easterns have very strong evidence of selection for viruses. So SIV is a really interesting one because it is not shared across all of the species. Now, if we turn this map, and what is important to say also is that we believe that Central and Eastern chimpanzees were infected around 100,000 years ago, probably a bit older than that. So if we turn this into a tree, what that would mean, given the divergence of these two subspecies, is that it is very likely that the common ancestor of these two subspecies was first infected, and that was a zoonotic transmission from two monkeys. I'll tell you in a second, and then that this infection has continued in the Central and Eastern chimpanzees through their whole evolutionary time. So I don't want to go into details, but the chimpanzee SIVs are recombinant between two monkey chimpanzees. So there was a chimpanzee that was infected by two different SIVs from two different monkeys. It recombined, and so it established itself so that it could infect chimpanzees and transmit. And that's how this zoonotic transmission happened. So we have a zoonotic transmission into chimpanzees, and then from chimpanzees into humans. Okay, so then what this means is that then they have not been infected for such a long time, and that might explain why we do see these signatures of selection for viruses in the Easterns. So now we can go with this hypothesis and ask specifically, do we have sets of genes that we believe should be under positive selection if SIV had been the main selective force behind those signatures that we observe? And there are two main sets of genes that we could use. The first ones are VIVs. VIVs are viral interacting proteins. Those are proteins that don't have an immune function necessarily, but they physically interact with the virus. And they have been shown to evolve quickly. They are responsible for adaptations in humans and in other species. And we believe that this is because viruses are such a strong selective force that the proteins that interact with them very quickly evolve under that selective pressure. There's no enrichment in VIVs for the Eastern chimpanzees. But we have enrichment in SIV response genes, which is the second category that we tested. And let me explain in a second what those genes are. So here are the vervet monkeys. The vervet monkeys are African primates. They are natural hosts. They don't get bothered by SIV. They are infected Each species has their own SIV strain. When they are infected, they start an immune reaction that changes gene expression, obviously, of immune-related genes and controls the virus. And this allows them to have very little or no health consequences, even though the virus replicates. It's just completely under control. Now, by comparison, you have macaques. Macaques are like humans. They are not natural hosts. They are naive species. These are macaques, Asian macaques. Now, when they are infected with SIV in the lab, they have also an immune reaction that also changes gene expression, but that doesn't control the virus, so that you end up having immunodeficiency. So the SIV response genes are the genes that show differential expression between the natural hosts and the naive hosts in CD4 cells, which are the cells that SIV infects. So these are the gene expression changes that experts believe are responsible for the fact that the natural host can control expression and the naive species cannot control expression. That doesn't mean that there are no other genes that are involved. That is completely possible, but these are very, very good candidates to explain the difference between naive hosts and natural hosts in how they can control expression. So this set of genes is what we call SIV response genes, and they are interesting because they are under constant positive selection in the vervet monkeys. So if you look at the genes that are under positive selection in vervet monkeys and constantly evolving quickly, this set of genes is one of the strongest ones, which is telling you that even though vervet monkeys have controlled the pathogenicity of the virus, it's still a strong selective force. And these are the genes that they are evolving in order to keep it under control. And this is the set of genes that show signatures of positive selection in Eastern chimpanzees. So basically what we see is that the Eastern chimpanzees have an excess of signatures of positive selection in genes related to immune-related functions and in SIV response genes. And actually, if you remove the SIV response genes, then the signatures of selection in the Eastern chimpanzees don't differ from those in the other subspecies, telling you really what we identified that was unique and unusual in the Eastern chimpanzees is due to this set of genes. So this is really exciting. What this is telling us is that by using population genetics, we can identify the genes that Easterns are evolving in order to control the virus. And to us, this was really interesting. Of course, this also opens a question of what's going on with the central chimpanzees. Central chimpanzees are also infected by SIV, probably as we believe, as long as the Eastern chimpanzees. They don't have very strong evidence of selection, even though, of course, there must be positive selection, but not as strong as the Eastern chimpanzees. But because we wanted to understand their evolution, we also ran the same enrichment tests to ask whether there was any category that was significantly enriched in the central chimpanzees. And there is one category that is significantly enriched, and that is chemokine receptors. And this is due to signatures of positive selection in 3 different chemokine receptors. And these 3, they are in a cluster, but we see independent signatures of selection for the 3 of them. They are paralogs of the co-receptors that SIV uses to enter the cell. So SIV uses CD4 to enter CD4 positive cells, and it needs a co-receptor. And this co-receptor it's a paralog of these genes. Actually, I mean, we know very much about HIV, how it enters cells. For SIV, it is not so clear because it is studied, but not to the same extent. But we believe that it is very likely that these genes have been under positive selection in central chimpanzees because they are co-receptors of SIV entrance. And if that is the case, what that would mean is that we have different mechanisms of adaptation in these 2 different populations to the same selective pressure, right? In this case, to SIV. And that is, of course, interesting from the evolutionary point of view because you have different adaptive mechanisms. But from the point of view of SIV, HIV, it also tells you that you can control or you can work into controlling the pathogenicity of a new virus in different ways, and potentially with similar consequences. I don't think we know because I don't think we have a very good understanding of whether the infection is identical for Central and Eastern chimpanzees. We don't know enough about that. And for good reasons, it's very difficult to do research on chimpanzees now. So, so this was a really nice evolutionary story for us. But of course, it opened the question of what's happening here. What is happening in the common ancestor, right? So if there was a zoonotic infection of the common ancestor of the Central and the Eastern chimpanzees, then we would expect to see also signatures of adaptation in that branch. And actually, I would argue those are the really interesting adaptations, because those are the ones that happen right after the zoonotic transmission. So they will tell you what do you need to change in order to reduce the pathogenicity of a virus that you just encounter, and that it's lethal, if we believe it probably would be, as it is in humans. So to answer this question, Harvey Power has done an analysis where she uses a different method. This is 3P-CLR, which is not based on allele frequency differentiation, but it's based on the signatures of positive selection on the allele side frequency spectrum. So if you have a selective event in the common ancestor of these two populations, then you would expect to see a certain signature in the allele frequencies, and that is exactly what this method does. I won't go into the detail of the methodology again, just to say that here we go to the tail of the empirical distribution and we investigate the genes that are there. So I cannot tell you whether or not we have very strong evidence of natural selection, because we haven't compared with simulations in this case, but here we're going with a different approach, which is our hypothesis is that we would expect to see adaptation to SIV in this branch, and we're going to specifically test the hypothesis. And I would ask you to not to tweet about this. We're still working on this. I will tweet it all the time, and I'm very happy to discuss papers that are published, but it's very difficult to have scientific conversations when people don't have all of the information. Anyway, so we do find sites that, so of course you go to the tail of the empirical distribution, you always have a tail of any empirical distribution, but we find that evidence that the sites that we identify have the expected signatures of positive selection. And I won't go into detail, but I can give you more detail if you like. They have exactly the patterns that we would expect if they had been under positive selection. Now, we can ask whether it is true or not that we see evidence of adaptation to SIV by studying their enrichment in gene categories. So what I'm going to show here is this is the false discovery rate for an enrichment analysis for a number of SIV-related categories. Yeah, let's just jump into this. So the only thing that I should say maybe is that this, if you see the star, that means that this significant at the 0.5%. So there we're studying again the same two categories, SIV response genes. So those are genes that change expression to control the virus. And here what we have is different thresholds of significance for the signatures of positive selection. And this is the strongest. So these are the strongest candidates. And then these are also, 5%, 0.1% and 0.05%. So SIV response genes have a very strong signature of positive selection among the strongest candidates of, they show a strong enrichment among the candidates of the strongest signatures of selection. And there are two different co-expression modules, I won't go into details, that also show some evidence of selection. These are the modules that show evidence of selection, for example, in the vermin monkeys. So these are sets of genes that change expression at the same time in the same manner. So we do have some evidence for SIV immune response in the common ancestor of the Central and Eastern chimpanzees. Now, for the VIPs, if you remember, I mentioned there's not very, very strong evidence of viral interacting protein positive selection in the Eastern chimpanzees. We have a lot of evidence of selection in viral interacting proteins in the common ancestor of the two species. And it's true for a number of viruses, but the two that have the strongest evidence across the different thresholds are influenza virus and HIV HIV1, 2, and SIV. So these are the ones that we expect to interact physically with SIV. So now we have, for the central chimpanzees, very strong evidence of positive selection on these two sets of genes. So if we think about this put together with what I told you before, what we have is in the common ancestor, so this population that probably first received the SIV virus, we have evidence for positive selection for VIPs, as well as other viruses, right? I mean, we don't pretend that this is the only selective force. There are other selective forces, but SIV seems to be a very important one, as well as SIV response genes. Now, SIV response genes also show evidence of positive selection in the eastern chimpanzees, so they seem to be a continuous selective force, but the VIPs don't show very strong evidence of positive selection later on, perhaps suggesting that they are part of the very first adaptations to new viruses. And then in the central chimpanzees, as I mentioned, we have the chemokine receptors. So we have some what seem to be different strategies of adaptation at different times and in different populations, but also some similarities between them, right? So SIV response genes, for example, are present in 2 different branches. So what we are interested in knowing then is, if you take this as one population, if we put together the information about the signatures of positive selection across time and populations, which is not an easy thing to do, but if we try to do that by combining the candidate SNPs and loci across all of these branches, what evidence of selection do we find? And we find evidence of selection in SIV VIPs, probably because of the strong effect early on, but some effect later on. But there was a very interesting category. There's only one pathway that is significant in the combination of all of these candidates of positive selection, and that's the Th1, Th2 activation pathway. And I won't spend a lot of time talking about this. It's a really complicated pathway that is used to CD4 T cells that are helper cells decide whether they will develop into Th1 or Th2. And that determines what type of immune reaction the organism is going to have. And there are in this pathway, there are 22 genes that show evidence of positive selection in at least one of these branches. And I believe there's 4 of them that have signatures of selection in multiple branches. And there are very interesting cases, like there are proteins that are encoded by 2 different genes, both genes showing evidence of positive selection. So what this is telling us is that this pathway has been targeted by positive selection in these populations constantly at different times. Now, the Th1, Th2 activation pathway is really important for immune function. It's very important for HIV and SIV for a number of reasons, including that it is active in CD4 cells, which are the ones that are infected by SIV. We also find evidence of positive selection in CD4, which is the protein that the receptor that HIV and SIV use to enter the cell. So what we believe that is happening is that this pathway that we had not identified and others had not identified, even though it's very important for SIV, it had not been identified as important for adaptation to SIV. To us, it seems like it's really a target that allows to reduce the pathogenicity of the virus. So just to conclude, SIV appears to be a very strong selective pressure in chimpanzees, even though they don't respond to it in the same way as humans. They have definitively some adaptations to it. We find evidence of adaptation to SIV only in the lineages that are known or predicted to be infected by SIV in nature. So we only find evidence of selection to SIV-related genes in Central Eastern chimpanzees and their common ancestor. We find evidence of different major mechanisms of adaptation at different times after zoonotic transmission and in different populations. And this is really interesting from the point of view of evolutionary biology, because it suggests that from the time of first infection, you have a mechanism of adaptation, and then you can have different mechanisms in different populations. Now, what we find is the Th1, Th2 activation pathway seems to be recurrently hit by selection across time and populations. As I said, it's just remarkable how many of these genes have been under selection. And this suggests to us that this is a critical biological pathway to reduce the pathogenicity of SIV and HIV. And of course, one can then start making comparisons between the chimpanzees and the humans and understanding which of these steps are really perhaps critical in the pathogenicity of HIV in humans. Now, to conclude, I would like to say that this whole process has got me really convinced that there is a lot of value in investigating genetic adaptations in species that are semi-natural hosts. So species that have been infected relatively recently and that are not perfectly adapted to the virus, because that allows us to identify the genes and proteins, the molecular mechanisms that are very important for the pathogenicity of a new virus. In this semi-natural host species, there has been enough time for genetic adaptations, and we can identify them so there has been enough time for genetic adaptations to appear, but we're still able to identify those first critical adaptations because we still have power to see them using population genetic methods. If we waited 20 million years, then we would not be able to identify them, even though they happened, because we have just lost the power to identify that. And let me just finish by thanking Harvey, Harrison, and Josh, who are the three people who have led this together with other collaborators. My group when we were in Leipzig, where we started this work, and then since we moved to London, where we've continued working on this and several other projects. And I think that I'll just stop there. Thank you for your attention, and I hope that you have questions.
Thank you very much. Outstanding talk. We certainly have time for questions. And I guess I'd like to begin with the one related to what you just closed with the Th1, Th2 work. Are the genes in that pathway actually observed to have or play any difference in response in human response to HIV?
Yes, many of them do. So many of them are known to have, for example, effects on the replication of the virus, or they are used by the virus in order to increase their replication while controlling the cells that they have infected. So many of them do. We did know that many of them are important for HIV. The one thing that I would say is if we ask, for example, the genes that we identify as important in adaptation in chimpanzees, and we ask whether they have been identified, for example, in association studies in humans, there's not very good correspondence between the two. And to me, what that suggests is that if you study humans, then you will learn the variants that you have in humans that mean that some individuals are more affected than others. With HIV, it's complicated because most people now are under treatment when they are studied, so it's not natural. But what this tells me is that those association studies in humans tell you something very important about variation in humans, but they don't tell you about which are the genes that are important to control the pathogenicity of the virus. So I think that those are two different ways to think about it.
And just a related follow-up, so pivoting in one direction as humans, then pivoted the other direction is in the vervets. Does that pathway appear to be a critical modifier in vervets as well?
I don't. That's a really good question. I don't know that. And actually, well, I know for a fact that in the study where they investigated the evidence of adaptation in the vervet monkeys and compared it to these SIV response genes, they didn't ask anything about Th1, Th2 because this is something that we have found. So that's a really interesting question. I don't know. We should explore that.
Cool. Questions from the audience?
Yeah, so do feel free to raise your hand virtually. We love to hear from you. You can do that by clicking reactions at the bottom of your screen and then virtually raise hand. I do have one question in the chat that came pretty early on in the presentation. This is from Bodan Arishkovich, who has a question about founder effects. And they're wondering, how does one use the term with reference to RNA viruses? Founder effects were initially described to describe the adaptation of multicellular organization to new, formerly empty environments or niches, but they're now being used to mean spillover of viruses from one species to another and how the virus moves. So they're wondering, is that a correct utilization of the term?
That's a very interesting question. So to me, as a population geneticist, to me, a founder effect is when you have a population where you have a population that colonizes a new environment. That's how I think about founder effects, right? So you have a subset of the original population that will generate a new population. So variation in the new population is a subset of the original population, and so on. So in some ways, I have to say that, I'm not a biologist, so I'm not sure that you should listen to me very much, and I'm sure you know more than I do about this. But in a way, I can understand that when you have a virus that has some level of diversity because it has been in a population for some time, and it infects a different population, that's a founder effect in the sense that the newly infected population the virus that infects the new population doesn't have all of the diversity that the original population had, right? So if you have, I think in a case like, I don't know, SARS-CoV-2, it's a very new virus, that's a different story. But if you have a virus that has infected, say, chimpanzees for a really long time, there's going to be some variation of that virus. And if there's one case of zoonotic transmission to humans, then that's a founder effect in the sense that HIV in humans does not have all of the diversity of SIV in chimps. I don't know if I'm answering the question because I'm interpreting it from a very pop-geny point of view.
Great, thank you for that. Other questions from the audience? We certainly have time for a few more.
Well, I'll ask another one while we're waiting for people to raise their hands or come off mute. It was a little less clear to me, is the reservoir for SIV ubiquitous across the entire 4 populations? I mean, in the monkey population, is SIV present just not in chimps, or is it isolated, or is it only present in reservoir in the monkeys where it's where there's resistance?
No, the origin of that question, I guess, is why don't the other 2 subspecies of chimpanzees are also infected? So I think there are, so what you're hinting at, which is a really interesting question, is, well, you have 2 subspecies where we don't see SIV in the wild. I don't think we can say for sure they have never been infected, but we know that we don't believe they have it now. So there's no doubt that they are in contact with SIV on a regular basis. SIV among monkeys is just everywhere. And actually the great apes, so chimpanzees and gorillas, are unique in not having their own strain of SIV and being infected relatively recently. So there's no question that they're in contact with it. I think that it is possible that they have never been infected in the same way as the other because it's such a complicated recombination event that generated chimpanzee SIV. So if you take monkey SIV, it can infect a chimpanzee, but it doesn't replicate. It cannot infect other individuals. This doesn't work. So it's not such an easy jump. As I mentioned very, very quickly, the chimpanzee SIV is a recombinant between 2 different monkey ones, right? So that was a really unlucky chimp that was they don't eat monkeys all the time, not all of the monkeys will have SIV. So it was just very unlucky that they just some individuals happen to eat at the same time or at around the same time monkeys with SIV, and that it could recombine and it had this effect. So I don't think it's very surprising that you don't see this happening all the time. The one thing that is less clear is because we know that there's migration between the Eastern Central chimpanzees and the Western Nigeria, Cameroon, why is there no transmission of the virus? Is it because just the populations that tend to migrate don't have very high prevalence of the virus, or is there something else? There's some people believe that maybe Western chimpanzees were infected and have somehow got rid of the virus. If I talk to biologists, they tell me this is not very likely to happen. It's very difficult to get rid of a virus that has been established in the population. But it's a really good question that we don't know. We hope that to be able to help address that. But from the evolutionary point of view, I can tell you is that we see no evidence of adaptation to SIV in the two subspecies that don't have it currently in natural populations. So we don't have it. Yeah, it's not impossible that it has happened, that they were infected in the past, but we don't see evidence.
Great, thank you for that. I want to check, I think Anne Pusey may have been trying to raise her hand. Anne, did you have a question?
Well, that was one of my questions. So that was very interesting to hear that answer. I mean, I just find it extraordinary, really, that because those West African chimps are eating a lot of monkeys, and there is SIV in the monkeys there. So I had wondered in the past whether they had managed to purge it somehow and would have genetic adaptations, but you're suggesting that you're not finding those.
So yeah, I think it's, but I wouldn't say. So we're studying this a bit more right now, and CD4, for example, have some changes in several of the branches that some studies have suggested it's possible that they have to do with old SIV infections. So I wouldn't say that it's impossible. What I'm saying is that our population genetics data doesn't tell us that that's the case, but this could change if we find evidence of positive selection. I think maybe because I know less about viruses than about population genetics, I really convinced when I talk to people who know much more than I do about viruses, and they tell me they don't think it's very likely that once a virus has established itself in the population, that the population would be able to completely remove it. So that it would disappear from the Western chimpanzees if they had a lot of selection, or sorry, if they had a lot of infection in the past. But now we're getting into something that is not my expertise, so I don't know. But it really is an interesting question. And actually, I mean, it's not that the Westerns have evolved to get rid of the virus. You can infect them in the lab, and actually they have been infected in the lab for many years for medical studies. So yeah, they can be infected for sure.
Yeah, I did have another point. I put in the chat, I don't think it came out very well, but we have been studying SIV at Gombe for the last 20 years with Beatrice Hahn. And we were able, because she developed these ways of assaying the presence of the virus through feces, we were able to track actual infection events and then keep track of what happened to those individuals. Some of them crashed and burned very early, and they did have they had symptoms like AIDS-like symptoms with no CD4 cells and so on. So I put some of those papers in the chat. And then we have 3 communities there. One community has declined. It was the one with the highest prevalence of the virus by far, and it's gone down to very few. And we think that that's at least partly because of the SIV. So at least at Gombe, and it's very hard to track pathogenicity unless you can keep track of individuals. And Gombe's been the only habituated population where we can keep track of individuals for many years. And we have found that, in fact, a lot of them die very quickly.
Yeah, so this is what I was referring to, or trying to refer to very quickly, where I was saying that hopefully you agree with me, that there was this view that the chimpanzees really didn't get AIDS and they were fine. And now the more people study this, the more we realize that there's health effects, and it seems effects on mortality and fitness. Yes. But what maybe I'm too far away from the literature, but what I don't get the sense from the literature is that you have the same reaction as you have in humans. So I would say that that it seems like they have some evolution and adaptation that has reduced the pathogenicity of the virus. But would you agree with that statement that they don't get AIDS as humans do, but that there are health consequences to SIV?
Well, I think we've had a couple of individuals who you could say they did have AIDS, yes. But on the other hand, we have some who are surviving quite well. But they do seem to die earlier. But you know, we're still following up, and it takes a long time to get a sample size to really get a good handle on how pathogenic it is.
Yeah, and that I have to say, I mean, thank you for that work. It's really, really extremely valuable, because before all of this, I mean, yeah, what we had is one chimpanzee who was infected with HIV and SIV and had AIDS or something, and that's not comparable to what you have in nature. But yeah, but what you're telling me is, yeah, it's the idea that I have that they're clearly functional health effects, which I think is exactly what you would expect if you have this kind of semi-natural host where not all of them get AIDS as you see in humans, but there's clear, they are not vermin monkeys. They suffer from SIV in some ways. Yeah, I probably have read all of the papers, but I'll make sure that I did. Thank you so much.
Thank you.
Great, thank you so much. So we are at the top of the hour, but Terrence Taylor has had his hand raised for a while, so try to squeeze in one more quick question. Terry, you can go ahead with your question.
Thank you very much indeed. Great presentation. I'm naive to the subject of using that in a slightly different context. And so I come from a health policy point of view and from human health. So just to help my understanding, your presentation is very clear, but the question really arose from your remarks about macaques and how they didn't have, they would suffer an immunodeficiency. Presumably the SIV, following your thinking, the survivors will provide the adaptation, or has there been a crash in the macaque population? Perhaps you're not working in that area, but and so has there been a crash or not? And is it just a question of they're not far enough along the timeline for evolution and order? Is it that simple? And this would help us think about the human population and reaction to smallpox, for example.
Yes.
But humans haven't had enough time for the evolutionary adaptation. Have I grasped the idea?
Yeah, no, it's an excellent question. So, and because I'm trying to say things very quickly. So the macaques are not, I don't know of any case of a macaque infected in nature, because in nature, SIV is in Africa. So the Asian macaques that I'm talking about, they have never seen the virus in nature. So the macaques that I know that have been infected are either because they're in zoos with other primates that are infected, or because they are part of medical experimentation. And they have been for a long time. And there, you know, if you infect a macaque with HIV or SIV, they have immunodeficiency. Very similar. I don't think it's exactly the same as humans, but similar. If there, I don't even want to think about this, but if there was SIV infection of Asian primates, I would expect that this would just go through the population and probably have a horrible effect, because, as you say, there's no time. I mean, there's no time for adaptation, right? So it's only through long-term adaptation that individuals that survive will carry those adaptations through time. So luckily, I don't think that has happened. But the one thing that is, we're very worried about the viruses that come our way, but our viruses also go the other way around, right? So chimpanzees, for example, the ones that live very close to human communities, they suffer from flu, for example. If HIV jumps into into Asian primates, that would be disaster. I believe very likely. I mean, it's hard to say exactly how well it would be able to jump from one species to another, but it's possible that it would be a big problem. I don't think about this very often, but it is really scary. And in the same, I mean, in the same way as in zoos and in the, I don't know, African sanctuaries, they have been extremely careful with SARS-CoV-2, right? So people who take care of bonobos, they have the strongest measures, because you really don't want to bring that virus into their communities. It's exactly the same.
Thank you.
Great, thank you so much. Well, we are past time, so we'll close now, but thank you so much, Aida, for that wonderful talk and discussion. Before everyone tunes out, I do want to quickly show that we now have our full Club EvMed spring schedule available on our website. So this is our talk you just heard, but below that we have our spring 2022 events starting on January 11th with Randy Nesse and Robert Perlman on control system failures in evolutionary medicine. So do check out the remainder of the talks. We have a really great semester coming up. And as always, I will send out a link to the recording soon, and we hope to see you at Club EvMed in 2022. Thanks, everyone. Bye.
Thank you. Goodbye.